So this is going to be my first teaching. We're going to go with lesson 2 of the unit plan that I created, and that's going to be the eukaryotic cell day. So we're going to be reviewing eukaryotic cells, doing the worksheet, and then doing reflective writing on it. So, the big idea is just understanding the structural components and the concepts of eukaryotic cells. So ultimately, they can be able to compare prokaryotic and eukaryotic cells. Once again, this is for a 10th grade general science, maybe a biology course. And overall, this will be the 3rd lesson of the unit. Good morning, class. Before we begin, I just wanted to talk about what we learned yesterday. So at this point, I would have a screenshot or a image projected of the following. That was the study guide that we did yesterday. I would just have that up on the board what we talked about yesterday. So again, good morning, class. I just want to review what we did yesterday, what we learned about the structure as well as the functions of the prokaryotic cell. So as we learned, there were some, there are some unique structures at the prokaryotic cell that you're not going to see in the eukaryotic cell, as well as structures in the eukaryotic cell that, were not found in the prokaryotic cell. So we'll get more into the development of eukaryotic cells later, but essentially it's hypothesized that eukaryotic cells evolved directly from prokaryotic cells, which would explain why they're so much more complex, as you will see in today's lab. So with that, we'll jump into it. From the age of dinosaurs to the dawn of man, the foundational blocks of multi-celled organisms have one thing in common: the eukaryotic cell, which we learned about yesterday. Throughout today's lesson, we will learn about the structures of eukaryotic cells as well as their function. By the end of class, you will be able to know and identify the structures as well as their purposes in the eukaryotic cells. And just to clarify, yesterday we learned about prokaryotic cells. Thanks for calling me out on that, I appreciate it. All right, so now we're gonna separate into our lab groups, just like we did yesterday, except we're gonna mix up the groups. So at this point, the students don't know this, but I'm arranging the groups based on sort of a limited ability grouping on what we observed yesterday. Students who are good with the microscope, they can be together. Students who had a better understanding of the content, that way I can focus more time and resources on the students that need more help. But that'll also help the students who seem to get it a little bit better, help them better facilitate conversation and get them thinking a little bit deeper, while I can focus my resources on helping the students that are still stuck at the surface level. So we're gonna move you two and you over here in your group. You, and you. You guys can take the group over there. You 4 can actually come over here, and then the remainers, I want you to come over here. So as you guys get settled, I have your microscopes set up with the first slide in. The first slide is going to be just a full shot of a eukaryotic cell, kind of macro version. And then we're going to go through each of the cells that are present in various eukaryotic cells. So I don't want you to get hung up on every cell containing these, because not every cell is going to have all of these structures. An animal cell, quote unquote animal cell, is not going to have a chloroplast. So if you see something, and we'll get to that later, but the important thing is I want you to be able to see what it looks like, and also understand that these are not fixed objects within the cell. They have the ability, they're floating around a little bit, so everything is kind of fluid. So just remember that when you're reviewing these slides. And as far as your lab journals, I want you to label, you can just put 4 bubbles on each page or however you choose to do it, but just make sure that you're labeling what you're viewing. So if it's, your first one should be eukaryotic cell. If your next one is ribosome, make sure you label ribosome, and then focus in and actually work on sketching that, so I can make sure that when I review them, I understand that what you were looking at, and we can go over it that way. So once again, here are your microscopes, and we'll go through and I'll be circling the room to facilitate any questions or help out with any troubleshooting. All right, let's get to it. All right, so during this phase, the students are going to be reviewing their slides and looking at them and recording what they see in their lab journals. So I'm going to walk around and ask prompting questions as well as clarify things. So some examples of what I might say are, the students are looking at it and I hear them talking about, they see the mitochondria. So I might come up and say, oh, that's awesome. What do you think the mitochondria does? How many of them do you see in every cell? If you see more of them in a cell versus less of them, what does that mean to you? And then we could have a conversation about the more mitochondria in a cell, the more energy it needs. So it's creating and converting that to ATP. Or we could see a structure of a chloroplast and say, okay, we see chloroplast in the cell. Well, what does that mean? Well, it means we're looking at an animal cell, right? A plant cell, a eukaryotic plant cell. And I think it's important for students to know that plant cells have both chloroplasts and mitochondria. That's something that I picked up in college level biology. I didn't pick it up in my general science in high school. So I think that's an important thing to clarify for students. Okay, so after the students have completed reviewing all the slides and documenting what they saw in their lab journal, we'd pick up from there and I'd say, all right, great job, class. We can return back to our seats and we're going to go over the class study guide. At that point, I would have placed on their desks the diagram that you can see here, along with the word bank that you can see here. So at this point, I would have that up on the projector or the smartboard or crudely illustrated on the whiteboard. Not much of an artist here. So at that point, I would just ask students, we would go down the list and I'd say, cell wall and pellicle. Great. So Jenna, we have the cell wall and pellicle here. Can you tell us what the function of that is? And, from there, we just go over it down the list. So, the cell wall and pellicle, they strengthen and give shape to the cell. So then we'd move down the list and, great job. Frank, what do the chloroplasts do? Can you tell us what the chloroplasts do? And he might not say, obviously, that photosynthesis, trapping light energy and formation of carbohydrate from CO2 and water. That's what the actual definition is here. His answer might be, it's how the cell takes up sunlight. That's great. We're looking for the students that create their own answers, so we can add that to our class board wall. It's important to build on not only academic language, but transfer that into real world language, because they might not remember, that a chloroplast, traps light and energy formation of carbohydrate from CO2 and water. They might not remember it that way, but they know that the chloroplasts are how photosynthesis happens, right? So, a little bit of knowledge there. Okay, so then, Connor, can you tell us what the cilia and flagella do? And he might say they synthesize proteins. Well, that's a great try. Actually, I could say that the cilia and flagella facilitate cell movement, or I could call on another student and say, Tammy, you're gonna take a try at this, and then that gives her an opportunity to correct it, as well as him learning from his mistake. Okay, so after we completed the class study guide assignment that we do in teams, we could move over to facilitating our word wall. So we might go word for word. We come to ribosome. Class, what does a ribosome do? The answer could be as simple as protein synthesis, or they synthesize proteins. So we could write that down and put it on our word wall. What I really like about this is it helps them draw connections. Like I said, it's their own words, it's the academic verbiage. We could put both on there. We could put a drawing for other things that would help students. So that's really great. And in the Almirod and Hattie text, they showed that the effect size for this is actually really huge. So we could come to something like, the nucleoid. Well, what is, we come to nucleoid and they give us the definition and say, that's great. Well, what's unique about the nucleoid is it's only found in prokaryotic cells. We know that the nucleus and the nucleolus is found in eukaryotic cells, but the nucleoid is only found in prokaryotic cells. So things like that are great because not only does it help them learn the vocab word, but it helps it put it into context. So as we're talking and going through some of these terms, it helps increase the learning as well as make new connections. All right, so one thing that we were asked to do for this assignment is to create an elastic activity, to help facilitate learning in the event that we have extra time. So class, we got a few extra minutes here in class. I want you to add to your lab journals and just write a reflective journal, 1 to 2 paragraphs about what you learned today about eukaryotic cells and what you learned about the structures that are in both cells versus unique to one cell. And tomorrow morning, I'll collect those and I'll review them over the night and give you guys feedback. All right, so as class started to wind down, we're getting closer down to the bell. I just remind students, class, what we learned over the last couple of days is something that we're going to be building on throughout the year. So I don't want you to just study for a test and data dump this. I want you to do your best to make connections. And if you have any questions, please reach out to me or discuss with a classmate. I really want you to be able to retain this because this is the basis of biology and most of the science that you're going to be doing at this level. And I also want you to think about and write a 1-page reflection on this. I want you to write a 1-page reflection on this. I want you to think about how prokaryotic cells reproduce and how eukaryotic cells reproduce. Not so much, whether it's, asexual or sexual reproduction. I want you to think about more how the process of the transfer of genetic material happens as far as the DNA. That's not going to be on this test, but I just want you to start thinking about that to build those connections because that's going to be what we're focusing on in the next unit. And I want you to grab a copy of this study guide on the way out. It's just a comparison chart. You can review it tonight. We'll talk about it more tomorrow when we dig into the nuts and bolts and start comparing prokaryotic and eukaryotic cells side by side.